charity organization, and our main goal is health and well-being for all. We do a lot of medical education programs for physicians, health fair, and we also do free clinics and free surgeries. Actually, the next free surgery program is on 22nd of July. We are going to do 8 free surgeries, and the free clinic is going on every week, Thursday in Downey. So Alapio continues to do good work even in this COVID era. If you like Alapio, please support us, encourage us, and if you want to donate, there is a website called alapio.org. And with that, I'm going to ask Dr. Jignesh Patel, he is our Alapio CME director, to introduce our speaker for tonight. Jignesh, go ahead.
Thank you, Bharat Bhai. It is my great pleasure and honor to introduce our very own Dr. Shikha Bose. She has been the director of cytopathology at Cedars-Sinai since 2002 and is clinical professor of medicine at both the David Geffen School of Medicine and Cedars-Sinai. She has a long, illustrious medical career. She trained at King's College Medical College in Lucknow and did her residency at Ames before moving to the United States. And in United States, she did 4 pathology fellowships over a period of 6 years. Fellowships in cytopathology, surgical pathology, fine needle aspiration, and breast pathology. So she has a very wide experience in pathology, many areas of pathology. She has published over 120 manuscripts and chapters, and she has lectured around the world on pathology. It's my great pleasure to introduce her. She will be talking about an update on the progress of the development of vaccines for COVID-19. So Shikha, please take it away.
Thank you, Jignesh, for that kind introduction. Hello, everybody. Good evening. So I have been charged with discussing the science behind COVID-19 vaccines. And towards this, I want to discuss, starting with the objectives to understand the mechanism of immune response in patients who develop COVID, understand the rationale of vaccine development, and also know the prominent vaccine candidates that are being developed. But before I actually go on to that, I want to give you all a very high bird's eye view of coronavirus. So the infectious agent for this disease is SARS-CoV-2, which is the short form of severe acute respiratory syndrome coronavirus 2. The disease is called COVID-19 from coronavirus disease 2019, since that was the year that we got slammed with it. The name corona comes from the electron microscopic image of the virus, which resembles the solar corona. If you see a picture, this is what we see with the naked eye during a complete solar eclipse. So that's where the name comes from, coronavirus. You can see the crown or wreath that surrounds the virus particle. There are several human coronaviruses or several coronaviruses that infect human beings. They are primarily divisible into 2 main groups. One, those that infect the upper respiratory tract. These generally called cause mild to moderate illnesses like the common cold, which are self-limiting. Then there are those which cause lower respiratory tract infection. These are very severe infections and they cause pneumonia and may even be fatal. We have encountered 3 such viruses, SARS-CoV from 2002 to 2004, which came out of China. The MERS from 2012, 2015, and 2018, which came out of Saudi Arabia. And now, of course, we have SARS-CoV-2, again, out of China. The bat is the natural habitat of this disease. and it is believed that since human beings do not interact very closely with the bats, there is an intermediate host that is responsible for transmitting this virus to the human beings. For SARS-CoV, it is believed to be the pangolin. The pangolin is otherwise known as the scaled anteater, and this is based on the fact that the coronavirus that is seen in the pangolin has 99% similarity with the human SARS-CoV-2. Few characteristics of the disease. Transmission takes place through respiratory droplets or airborne, tiny airborne droplets. Question of whether there is fecal-oral transmission is still questionable. The R0 or the R0 of this infection varies from 2 to about 4. R0 being the number of people that one infected individual can transmit this virus to. The infectivity 2 to 4 is somewhat mild to moderate. If you compare it, just to give you an idea, measles has an R0 of about 12. So that is really highly infectious. The incubation period is about 5 days. Infection can be either asymptomatic or symptomatic. The disease, based upon the recent testing numbers, it has been estimated that about 40% of individuals are asymptomatic or pre-symptomatic. Symptomatic individuals, 80% of them have very mild disease. 20% will require hospitalization, of which about 5% are critically ill. The infection and case fatality rates, as you see, are there. This is a diagrammatic representation of the coronavirus. It comprises of a central core, which is the viral genome. This is a single-stranded RNA virus. This genome contains all of the genetic codes that are required for replication of the virus and forming or producing all of the proteins that are required. The core is surrounded by a lipid envelope, and on the lipid envelope are several proteins that are embedded. Of these different types of proteins, the spike protein is of most interest because this is the protein that targets human cells and mediates the host cell attachment and entry into the host cell. There are about 77 to 80 spike proteins in one virus particle. So this is a diagrammatic representation of how the SARS-CoV-2 enters into the host cell. As I mentioned, the spike protein is the one that interacts with the host receptor. The receptor that it interacts with is known as the angiotensin converting enzyme 2 receptor. Once the spike protein interacts with it, it gets activated. It involves a protease that is present in the cell membrane. This then causes lysis of the cell membrane with fusion, and then that leads to a small pathway, opens up for the virus to enter. Once the virus enters into the cell, the RNA is liberated and it starts virus replication within the cell. It takes over the cell machinery, produces viral proteins, which then assemble to form new virus, which are then released into the surrounding tissue. Now, this process is detrimental to the normal cells because it's producing viral protein, and all of the functions that need to happen do not happen for normal cell maintenance. So the cell undergoes what is known as pyroptosis, which is an inflammatory type of programmed cell death. When this cell dies, there is liberation of various toxic chemicals into the surrounding, which then attract inflammatory cells to the area. Inflammatory cells in the form of T cells, macrophages, monocytes, these then secrete various chemokines and cytokines, which are interferon gamma, interleukin-6, and others. And this is what is known start of the innate immunity. So innate immunity is the first response of the body to any microbe that has not been previously exposed to the body. So this is our first line of defense. The NK cells, the interferon gamma that is produced, results in the production of NK cells, which are cells then that eat up the virus containing cells. In the event that the innate immunity is not sufficient for clearing the virus, the adaptive immunity comes into play over a period of days and weeks. There are two arms to this adaptive immunity, one of which is known as the humoral immunity, which comprises of lymphocytes, which are a specific type of lymphocytes that on maturation result in the production of antibodies against the virus. Now, this adaptive immunity is it specifically targets the specific microbe, whereas innate immunity is just a general sort of immunity that since the body has not seen the antigens of that virus, it is sort of a general attack on the microbe. The other arm of the adaptive immunity is T cells or cell-mediated immunity, which is brought about by T cells, which when they mature form cytotoxic T cells, and these cytotoxic T cells then are able to attack the virus containing cells. So in an individual who has a healthy immune response, what happens is when the cells are released, the adaptive immunity, the humoral immunity produces antibodies which inactivate the virus that is freely available in the circulation or in extracellular tissues. The cells that contain the virus are then eliminated by cytotoxic T cells. Macrophages are responsible for eating up all the cellular debris. There is minimal tissue damage and the infected cells are cleared. This is probably what happens in individuals who have asymptomatic infection or have very mildly symptomatic disease. In individuals who have a defective immune response, there is an excessive amount of inflammation that is produced. The reason for this is not entirely clear, but there is excessive inflammation. There are numerous inflammatory cells which secrete whole host of different types of cytokines in an effort which is way out of proportion to what is required to clear the virus. And as a result of that, there is not only damage of the cells containing the virus, but there is damage to the cells. Okay, so there is damage to the normal cells and the normal tissues of the body. This process where there is an excessive amount of inflammation with excessive cytokine production is known as the cytokine storm and happens when there is severe COVID. In addition to that, as I mentioned, that the ACE2 receptor is the receptor that the virus uses to gain entry into the cell. Now, ACE2 receptors are present in large amounts in the lung, in the GI tract, and in blood vessels, in the kidney and the heart. So these organs are particularly susceptible to the virus. The blood vessels, when they are injured, lead to the formation of microthrombi. The lungs, with the excessive exudation of fluid, gives rise to acute respiratory distress syndrome, which leads to reduced oxygenation in the tissues. There is another interesting phenomenon that takes place in some virus infections. Coronavirus is one of them. And in addition, the Zika virus and dengue fever also see this, which is known as antibody-dependent enhancement. Now, the body immune mechanism results in the production of antibodies which are supposed to neutralize the virus. So in the case of coronavirus, any antibodies that are produced against the S protein will neutralize the virus. But as I showed you, there are other proteins which are present on the surface. These are antibodies that are produced against those antibodies are known as non-neutralizing antibodies. And these non-neutralizing antibodies, for whatever reason that is not clear, they enhance the entry of the virus into the cell, and they enhance the production of excessive virus. So this is another thing which exaggerates the disease process, and something that we have to keep in mind when we're looking at vaccinations. Diagnosis of the disease, the most specific test is the molecular assay by RT-PCR, which detects the viral genome or the RNA. The virus can be detected from a few days prior to the onset of symptoms and may be present in nasal secretions, in pharyngeal swabs, sputum, bronchoalveolar lavages, blood, feces, and even urine specimens. Towards the end, later stages of infection, the viral loads are variable in the upper respiratory tract especially, and this can sometimes give rise to false negative results. It is believed that the virus shed at the later stages of infection are not really infectious. There are some other rapid test, point of care type of testings, but the sensitivity and specificity of those tests are not as good as the molecular test. Now, antibody testing, it is recommended that antibodies should not be used to diagnose disease or even whether a patient has immunity or not. And that is because of this main reason. There are some studies that say that 30% who recover fail to develop antibodies in their serum. The antibody testing at present is primarily recommended for population studies, for screening of potential donors for convalescent plasma, and the effectiveness of vaccines. Here is a graphic representation of the infection. So notice that, say the onset of symptoms starts at day 5. The virus, the blue line, is represented by the presence of the virus in the nasopharyngeal swab. You can detect the virus about 5 days before. It reaches peak at about 2 weeks and then generally disappears about 4 weeks. Sometimes the virus has been known to persist to even up to 6 weeks. IgM and IgA are the initial antibodies that are developed or seen on serologic testing. This indicates active infection. They appear at around 7 days, reach a peak at around 2 weeks, and disappear about 3 weeks. IgG develops a little late, around 14 days. This is supposed to be the most specific for the infection. It reaches a peak about 3 weeks or 4 weeks, and then persists for an unknown period of time at this time. There have been some recent studies from China and UK from recovered patients who, like I mentioned, may not show antibodies or the antibodies may not even last for a long period of time. It has been seen that after 3 to 8 weeks, the antibodies just disappear. Okay, so this is just to give me notice that I have to now change gears and move on to my actual topic of vaccines. So what is a vaccine? A vaccine is a biologic preparation that contains an agent. Previously, it used to contain portions of the virus which were inactivated or reduced immunogenicity, but now any agent resembling a disease-causing microorganism which stimulates the body's immune system to recognize the agent as a threat and destroy it, not only at the present time, but also in the future if the body encounters that agent or any microorganism associated with that agent. The other term that we have to familiarize ourselves with is herd immunity. What this means is that it is a protection of a community or a state or a country from the spread of a contagious disease. This herd immunity occurs when majority of the population is infected with the virus, either by falling sick or by vaccination. SARS-CoV-2 infection, it has been estimated that 70% of the population needs to be immunized against the virus in order for the community to be saved from this disease. There is a huge effort going on for development of vaccines. We still don't have a wonder drug, and everybody is now focusing on vaccines for prevention of this disease. And for us to get back to a normal life, public enterprises, academic institutions, private industries, all countries all over the world are collaborating. North America is leading the effort. And as you will notice, the private and industrial component is really good. The federal government has created a public-private partnership to facilitate and accelerate the development, manufacturing, and distribution and acceptance of COVID-19 vaccines. The aim or the charge of this is for not only for vaccines, but also for therapeutics and diagnostics. It's named as Operation Warp Speed, or OWS. The aim of this at the present time is to at least deliver 300 million doses of a safe, effective vaccine by January 2021. Now, vaccine development. The normal paradigm or the traditional paradigm for developing a vaccine takes multiple years. Initially, there are different phases in the development of the vaccine. Initially, there is identification of the target and preclinical trials on maybe mice, monkeys, chimpanzees. Once that has shown to be effective, it moves on to humans. The phase trial, which is in a small group of individuals, maybe 10, 20, 40, 50. And this is initially for the purpose of determining dose of the vaccine, whether there is an immune response of the vaccine, and this is usually tested on normal, healthy human volunteers. Once this stage is successful, vaccines can progress on to phase 2, which includes hundreds of patients, people who are given the vaccine. And again, at this stage, instead of just the normal healthy volunteers, different volunteers at different ages, children, pregnant ladies, older individuals are also tested again for whether what the accurate dose would be, what the effects are, and whether or not there is an effective immune response. Phase 3 is the final phase of testing the vaccine. In this stage, thousands of individuals are given the vaccine and compared to a group of individuals who are given a placebo, and they are then, the vaccine is given in an area where the disease is rampant, and the test is to see whether those who have been given the vaccine are protected from the disease or not. And then the last stage is the licensure stage, where the vaccine is licensed, provided all of these phases have worked out properly. In an outbreak or in a pandemic like we are in now, this whole entire process of multiple years is compressed. Target identification, preclinical trials are compressed. The labs are working round the clock to come up with results. Phase 1 and phase 2 are generally combined. Phase 3 goes on. And at the same time, even before we decide which vaccine is effective, whether it is safe or not, manufacturing is ramped up even before the process of knowing whether there is a vaccine or not. So the outbreak paradigm is completely compressed. And if we look at some of the vaccines that are now at the forefront, from the time we were diagnosed, about 5-6 months, we have vaccines that are in phase 3 trial. That means we have compressed several years into a few months, which is remarkable. A whole host of different types of vaccines are being developed. These are some of the types, prominent types, the virus vaccines, which include whole viruses. They may be either inactivated or weakened. There is the viral vector type of vaccines, which means that portions of the SARS-CoV-2 are inserted into a carrier virus. Instead of using the whole virus, we use only a part of the virus, which is inserted into a carrier virus and then delivered. Nucleic acid vaccines, these are relatively new type of vaccines where the DNA or RNA molecule that codes for the protein is created and then injected into the cell so that the cells that nucleic acid and then protein base. So instead of giving the code for the virus or the spike protein, the actual protein is injected. Now the race of the vaccine, race for a vaccine is on. And now I will go over some of these vaccines in a little bit more detail and discuss the prominent ones in the field. So whole virus vaccines, as I mentioned, is conventionally of two types: weakened vaccines or inactivated viruses. This was the conventional type of vaccines. That weakened virus one contains a vaccine that has been passed until it picks up mutations that make it possible, less to cause disease. That means the virus loses its replication properties. These viruses actually create a strong and long-lasting immune response, which provide lifetime protection. They're contraindicated, however, in immune-suppressed individuals. The inactivated virus vaccines are those which are rendered uninfectious, but still maintain their antigenicity. They are made uninfectious by treatment with some chemicals like formaldehyde or heat. But this process of making them uninfectious also reduces their immunogenicity. And therefore, these viruses are less immunogenic as compared to whole virus vaccines. The advantage is that this stimulates both the arms of the immune response, the humoral as well as the cell-mediated response. The disadvantage is that because they're live or weakened viruses, they need extensive safety testing. They may cause antibody-dependent enhancement, and transportation and storage is always a difficult problem because they need cold temperatures. These are some of the whole virus vaccines that are leading the field. China is foremost in this field, and not too left behind is our own Bharat Biotech from India, which just actually started phase 1/2 trial yesterday. They created a little bit of controversy because they said that by August 15, they will have the vaccine ready. And the time was really not very reliable or realistic. The Chinese vaccines are both in phase 3 now. They have reported initial good results. However, China is facing a little bit of a problem now with their phase 3 trials, because as you know that the incidence of the disease is now reduced in China. So they don't have those huge numbers of patients or population where they can try the vaccine out. So they have to go out and collaborate with other nations. They're working with United Arab Emirates and also in Brazil. Then moving on to viral vector vaccines, the spike protein is the one that is being used for the majority of the vaccines, not the whole vaccines, but for all other vaccines. The spike, everybody is mostly concentrating on the spike protein because prior experience with the SARS-CoV has shown that the spike protein is the one that is the most immunogenic, and antibodies against the spike protein are responsible for neutralizing the virus. So in the viral vector vaccines, the spike protein is inserted into the viral genome of the carrier virus. The carrier viruses are generally live viruses of low pathogenicity. That means like adenovirus, which causes a common cold, vaccinia. Different companies use different types of viruses. These again can be of two types, those which are weakened, such as measles, or those which are non-replicating, such as the adenovirus. This is the carrier virus. All of these viruses, you have the spike protein that is inserted. Once it gets into the cell, the spike protein is expressed by the viruses and an immune response is generated. The advantages are that specific genes can be delivered instead of using the whole virus. You can select out the genes that of interest and then insert them into the carrier vaccines. These vaccines are very good because both the humoral and the cell-mediated immune responses are seen. They are safe and they are more efficacious. The dose required is low, they enable large-scale manufacturing. The main disadvantage is that the pre-existing immunity to the viral vector may attenuate the immunogenicity. Like I mentioned to you that the adenovirus, which is a common cold virus that is used as a carrier, a lot of people get common colds, and in one Chinese study, they found that 50% of the individuals had some immunity against the common cold. So that reduces the immunogenicity of these. And rarely there is a risk of integration into the host genome, which might lead to some rare diseases. The common viral vaccines, China is one. This is very interesting because this CanSino Biologics has, on June 25th, was approved by the Chinese military as a specially needed drug, so that this vaccine is now being used or being given to Chinese military people. However, the other vaccine, the Oxford University vaccine in collaboration with AstraZeneca, is a chimpanzee adenovirus vector, which is in phase 3 trial right now in Brazil and South Africa, and phase 2 and 3 combined trial in England. They anticipate that they may be able to deliver emergency vaccines by October, and they have a huge capacity for ramping up their production. These are two other companies, Johnson & Johnson and Merck. Johnson & Johnson is using the adenovirus 26, which is not as commonly encountered as the adenovirus 5. Merck is using some different vesicular stomatitis virus and the measles virus. This platform for both of these are both in preclinical stage, and phase 1 and 2 are expected to start shortly. The only interesting feature about this is that both the platform that both of these companies are using have been approved previously for an Ebola vaccine, so we know that their platform works. This is another viral vector vaccines, and the reason I put it in here, both are again in preclinical stages. They are using the adenovirus 5 as their carrier. These are two very interesting because they have somewhat of a different product. The Vaxart is creating an oral tablet. So intranasal vaccine, Immune is producing an intranasal vaccine. The advantages of these vaccines is, one, they're stable at room temperature, so there is ease of storage and they can be easily distributed. But more importantly, they provide a broad immunity. Because this is an oral tablet, where they produce a broad immunity in that because they go through the intestine, so they produce local mucosal immunity. What is more interesting and what I really like is about this Altimmune, because it is producing an intranasal vaccine, and that means that the virus is released in the nasal cavity, and that's where the normal infection takes place. So if they are successful in producing immunity, they would be able to produce immunity not only in the nasal cavity, but also in the rest of the body. This is important because it has been seen in some of the vector vaccines that when they were administered to chimpanzees, that the chimpanzees, when they were challenged with viruses, they still got the infection. The viruses could still be isolated from the nasal cavity, although serious infections did not take place on those animals. Okay, moving on, nucleic acid vaccines. This is a relatively new process. The RNA or DNA vaccine, the spike protein is created in the lab. So they're not actually taken from the virus, they're created in the lab. The RNA vaccine is encased in a lipid nanoparticle, whereas the DNA vaccine may be put into plasmids. Plasmids are small DNA molecules which are capable of proliferating in the cell extrachromosomally. The advantage of these nucleic acid vaccines are that they do not require genetically modified living cells. It is possible to combine multiple RNAs, so multiple spike proteins or different proteins into a single vaccine. They are faster and cheaper to develop. They're safer because the stuff that is inserted is non-pathogenic. The main question, however, remains that there is no vaccine to date that has been licensed for clinical use using this technology. Also, it is not clear at this time whether the antibodies produced by this vaccine will be protective and whether they will stimulate a cell-mediated immunity, or will it just be a humoral response. The nucleic acid vaccines in the field are those by Moderna and BioNTech and Pfizer. These have recently published, the Moderna has recently published its results from phase 1. And phase 3 is expected to start on July 27th. For Moderna, this is in collaboration with the NIH, which is the National Institute of Allergy and Infectious Diseases, of whom Fauci is the boss. And BioNTech and Pfizer are also expected to start their phase 3 trial soon. Both have shown very good results in their initial phase 1. We are still waiting for the published results. We have not seen it in a journal as yet. Nucleic acid vaccine, Inovio, is another interesting one. What I told you before, we're all using the RNA technology, whereas Inovio is using the DNA vaccine plasmid technology. The good thing about this is at the same time, it is also preparing to initiate a phase 2 vaccine trial for MERS. And they have shown initially that both humoral and T-cell responses are present when they use this vaccine. CureVac is an interesting thing. It's still in pre-trial positive preclinical results. The reason I put this in is because it's creating apparently a mobile molecular printers which will create these vaccines. So they'll produce RNA, and they can be these like little mobile labs which can be transposed from one place to another. And Tesla, our favorite Elon Musk, has offered to help them to do this. He has offered to build molecular little mobile printers for this company. Moving on, protein-based vaccines. So instead of using the DNA and RNA, we use the spike protein, which is created in a lab. With that, what is required is an adjuvant is required to stabilize these proteins, and these are then directly injected. Virus-like particles are empty virus cells which have no RNA inside, but they contain all of the antigens, and therefore they can produce an immune response. The time to establish cell lines needed for manufacturing these vaccines is much longer than what is required for nucleic acid vaccines. The adjuvants may be limited, inhibiting the protein. You cannot do this because the protein is otherwise does not survive. There is, however, abundant commercial experience. There are several licensed vaccines which are available, which have used this technology, which is the hepatitis B vaccine, the human polyoma virus, varicella zoster, and influenza. So this technology is a known technology, which has been known to succeed. The protein-based vaccines, Novavax, is initially in phase 1 and 2. It is using spike protein, which are embedded in a nanoparticle. Phase 3 is expected to start in September. They have a flu vaccine in phase 3 based on the same platform. Medicago has recently, this is a company out of Canada. This company recently started phase 1, I think a couple of weeks ago or months ago. They have a very interesting product. They are creating a plant-based protein vaccine. What they're doing is they create virus-like particles, and what I found was very interesting is that they're using the tobacco leaves to create this, and they have a proprietary technology. I couldn't get the details of what the technology involves. They have, in fact, influenza vaccine out of Canada using the same technology that has completed phase 3 on 25,000 people and is at present in review for licensing for the influenza. So that platform is also a good confirmed platform that it works. University of Pittsburgh has a very interesting product in preclinical development. They have created a skin patch which is tipped with 400 tiny needles made of sugar. When the patch is placed on the skin, the sugar, the needles dissolve, and the virus is delivered into the body. This is still very much in the preclinical stage, but I found that is very interesting, so I put in there. With all of these vaccines that are being developed, there are still some huge questions that need to be answered before we find an effective vaccine. The first and most important question is, are neutralizing antibodies protective against the disease? We have seen that the antibodies are not present in the serum for a very long period of time. There might be individuals who have recovered from the disease but do not have antibodies. Are these individuals protected by antibodies, or have they responded to the virus by the cell-mediated response? Those are all questions that are not known at this present time. Does the quantity of antibody response determine efficacy of the virus? All of the viruses, all of the vaccines that are being at present tested are all being tested in phase 1, phase 2. They're all being tested in the lab. Volunteers are being given the vaccine. The antibodies are taken off from the serum, and in a lab petri dish, they see whether those antibodies are effective in neutralizing the virus. What will happen in the real world is still not known. How long do antibodies or the protection last? So if you get an injection, you get antibodies, you're immune to it, how long will it last? There is a whole variety of immune responses. The coronavirus, which causes cold, you get antibodies that protect you only for about 6 months. And after that, you're able to get cold again, catch a cold again. There are virus vaccines, antibodies that give you lifelong immunity. And then there are diseases where antibodies are produced, but they do not give you any, they do not protect you from those diseases, like HIV, for example. And lastly, are there any serious side effects to the vaccine, like antibody-dependent enhancement? That is another thing that we will have to find out. Will COVID-19 incidence somewhere in the world be high enough to be able to show that the vaccine can protect people sufficiently? A problem that happened with the SARS and the MERS viruses, the disease disappeared before they could develop a vaccine. It's happening now in China, where the numbers have gone down. They don't have enough people on whom they can do a phase 3 trials. So fortunately or unfortunately, we are not in that stage as yet. So the OWS has partnered with more than 18 promising candidates, have been chosen from 100+ vaccine candidates to accelerate development. There were 5 finalists who were sort of declared on June 3, AstraZeneca, Pfizer, Merck, Johnson & Johnson, and Moderna. Additional funding was provided to Vaxart and Novavax. They have chosen 3 to fund phase 3 trials: Moderna, the University of Oxford vaccine, and Johnson & Johnson. Pfizer has been awarded a fast track by the FDA recently. Pfizer has not taken any funding from the government because they say they have enough funds of their own. And collaborating or taking funding from the government delays their process, so they think they can get to it quicker by themselves. The requirement by the government to actually approve a vaccine is that the sponsor would need to provide data that their vaccine is at least 50% effective against COVID-19 in order to be authorized for emergency use. This is the coronavirus vaccine tracker, which gives you all the numbers of vaccines that are in play at the present moment. There are 135 or more than 135 vaccines that are being tested in preclinical phase. In phase 1, there are 15, phase 2, 11, 4, and 1. These are the vaccines. This was the one that was approved by the military. These are the vaccines that are in phase 3. This was the AstraZeneca. This is the Chinese vaccine, the 2 Chinese vaccines. And I forgot to mention, this is out of Australia. Murdoch's Children's Institute is trying to repurpose the BCG to see if this is effective against COVID. And they are in phase 3 trial too. Moderna and BioNTech have recently completed phase 2 and are expecting to start. Moderna has given a date of July 27th, and BioNTech has said in July, later part of July. The other important part to note is that from previous history, any vaccines from phase 1 actually go to approval. It's only 16% of vaccines will actually move from phase 1 to approval. Vaccines from phase 2, only 24% of vaccines will actually move through the phases. So again, that is something to remember. Not all of the vaccines that are being tested will go all the way up to approval. So that is my talk on vaccines. We are still in the early phases. We will conquer this horrible disease. But in the meantime, everybody, please keep safe. And I think I am open to questions.
Doctor, how come China is able to contain and bring down the problem epidemic fast? They don't have a vaccine. They don't have therapeutics. What is the reason?
They have very strict rules about locking down.
That's it.
Besides that, is there anything else, medically speaking?
No, I don't think so. I think that was the most important thing, that as soon as they found the virus, as soon as they discovered that it was going to be a dangerous one, they absolutely locked down everything, and very strict lockdown rules. And
Yeah.
you know, the locking down is very important. If you notice in California, our numbers were really good in the beginning when we were all locked down. And once the lockdown started being relaxed, the numbers have started flying up.
Yeah, we see a lot of people in Newport Beach, you know, we're not wearing masks, going to the beach, and it's so lax.
Yes, that is true. That is true.
But excellent dissertation and talk, very lucid and very.
Thank you very much.
Any next question? Who has the question?
Can I ask you a question? This is Padma.
Yes,
Go ahead.
And Shikha, first of all, it was a very good talk.
thank you.
I really congratulate you on that. Second thing is that, you know, this is not related to the vaccine, but, you know, in your readings, did you come across, I think there were some studies which said that, you know, people who are eating meat have a higher incidence than others. Did you come across anything?
No, not really. I think India is made up of a lot of non-meat eaters, whereas the US has a very prominent population of meat eaters, and we're both in dire trouble right now.
Hmm.
Okay, next question.
Can I
And
ask
who
you-
has the next
I have a
question?
Can I ask, when the
Shikha,
vaccine comes in the market, what are the side effects from the vaccine and how safe is it?
So that safety is still has to be tested in the phase 3 trial, but from the initial 2 companies or 3 companies that are in phase that have completed phase 2 and have published their results, the vaccine, the doses that have been selected are very safe. They cause only mild fever, some problems a little bit, not very serious. Nothing serious has been detected as yet. But that is the thing, as yet. You know, the numbers of people that have been tested are really very small, hundreds.
Yeah, because I read somewhere that it may be some side effects. Yeah.
Yeah, so we still have to test the vaccine in thousands of people before we know for sure.
Okay, thanks.
Okay, next
I
question.
have a question. Thank you, Shikha. 300 million vaccines. Is that going to be enough?
Well, they're going to start. They're going to start with 300 million, and then they're hoping to ramp it up to billions.
Okay.
Shikha, thank you very much. It was a wonderful talk. I think you gave a very comprehensive talk about the development of vaccines and also potential that we're really not seeing anything that is going to be definitive down the road. Most important factor is prevention. What I want to ask you about is how much does increased viral load affects the pathogenesis of the disease? That means that if there's an increased viral load. Now, second part to it, a lot of facilities in India are using, are having nasal lavages, especially people living on the frontline workers, thinking that they're clearing up all the viral loads on their noses so that they can work, and they have found that it's pretty successful, especially in Pune. So can you comment on that, please?
Okay, so as far as the viral load is concerned, yes, disease severity can sometimes be related to the viral load. People who have severe illnesses have a higher viral load as compared to those who are asymptomatic or not, or mildly symptomatic. And as far as your nasal lavages are concerned, I have not read any comparative studies where they have compared it with a group who didn't have nasal lavages to say whether that is really helpful or not. I really don't know about that.
I have a question.
Sure.
Shikha, excellent and very comprehensive.
Thank you.
Really enjoyed the talk. Has there ever been a documented true reinfection of a person who has had previous COVID-19 infection?
Yes, there has been a documented reinfection. There is a study out of China that I looked at where, the initial study, and there are some reports out of UK, the numbers are small, but the study from China, they studied 651 patients who were classified as recovered. And I couldn't get, okay, so there were 651 patients who were classified as recovered with a median follow-up 48 days. They found that 3% of patients tested positive on retesting. Now, I don't have the details of what the interval was between the retesting and the original testing. Out of those who tested positive, again, 65% had no symptoms, and there were only a minority, 35%, who had at least one symptom with active COVID. So, the number is really small, and I don't have the details because, as you know, the viral antigen can persist in the wherever, in the nasopharynx for a long period of time. And towards the end of later stages of the disease, you take the viral sort of the release of the virus is very intermittent. So you will have sometimes that is positive and sometimes that is negative. And if you do a test at that time and you catch it, it's not there. Sometimes you could, so it could be falsely negative. So, you know, there are a lot of issues that have to be sorted out with this before we can say that infection is not possible. There have been other studies that are now being shown that the viral antibodies disappear in a short period of time. Now, if that is the case, then that would mean that if the antibody is gone, that means that the immunity has gone too. We don't know. We don't know that. So if the immunity has gone with the disappearance of the antibody, then it is possible that we might get reinfected. But again, there are a whole lot of questions. There are some reports which are, I don't know how reliable they are, and what we still don't understand the entire life cycle of the virus. So as long as that is not known, even if there is a person who has been exposed and recovered from COVID, I would suggest please wear your mask.
Shikha, this is Smith here. I have a quick question for you. Thank you for the great talk, of course. So, of course, a lot of things are unknown, and most of us know that vaccine is not a cure. When we look at the life cycle of the vaccine, so for example, the flu season, we go once a year. In your best estimate, how often do you feel or do you think we would be looking at a revaccination? And I know you just said, of course, we don't know what the life cycle is.
Right. So the, you know, if you look at coronaviruses by themselves, there is the coronaviruses that cause common cold, which have a very limited, you know, protection produced after you get the disease. So if you give a virus with the coronavirus for common cold, you would have to give it every 6 months or a year, or even more, or somewhere around that. But if you take the example of MERS and SARS-CoV, the original one, SARS-CoV original one, after the disease, there was a protection for at least a year or 2 years. MERS, there was a protection for 3 years. So now in this range is somewhere that I am thinking that we would be falling in with SARS-CoV-2.
Bose, I have a question about BCG.
Sure.
So we have a clinical trial going on here in the US with the BCG vaccination.
Mm-hmm.
In India, and most of the people who are on this forum right now has been BCG vaccinated.
Correct.
Despite this thing, the incidences in India are going up. We don't see any difference in the trend between India or other countries.
Right.
And with that observation, why still have this clinical trial going on? Can you shed some light on?
You know, I don't know. The thing is that with the BCG vaccine, you know, we get the BCG vaccine, and it is, my understanding is that the immunity from that one dose that we get reduces over time. So in about 10, 12 years, the effect is lost.
Yeah, Shikha, you know, I'm actually a participant in the BCG trial.
Wonderful, then you can answer this question.
And the thoughts behind the BCG vaccine is that it's one of the few vaccines that actually stimulates your innate immune system. So it is not a vaccine against a particular pathogen, but it actually stimulates the innate immune system that Shikha so nicely described. That is your first response to an infection, and which is a non-specific response. Now, it is felt that if you've had the BCG vaccine when you were young, then it's probably not persistent. And so in this clinical trial, patients are being randomized to either a placebo or being vaccinated again. And you can get vaccinated even if you've had it previously. So that's what they're looking at.
Yeah, hi, Ajay here. So,
hi, Ajay.
So Shikha, very elegant talk, very comprehensive, and learned a lot. So early on during the course of coronavirus, there was this thought that there is the A, B, and C types of diseases. And so Like, for example, the New York disease was much more severe than the California disease because the virus was of a different kind. And I don't remember which was A and which was C. So has that, how has that panned out, and how has that flushed out? Does the science still support these different types of viruses, because vaccination would then be specific to that particular type of strain of virus, if you will?
Yes, so I would like to share my screen just one more time, because I did read about this, and I thought I would put in, but my talk was getting too long. So I have the slide here. Sorry. Okay, so this is from a recent article in the Journal of Cell, which says actually that they have now decided that there are two types of SARS-CoV-2, one which has a spike G614. This is a spike protein. The G has replaced the D as the dominant pandemic form. This is consistent with increase of the G614 at the regional levels and may indicate a fitness advantage. The G is associated with lower RT-PCR counts, suggestive of higher viral loads in patients, although not with increased disease severity. So the G has taken over what originally started off as D, and I think the D614 spike protein, so this is the spike protein at position 614, the D has been replaced by the G. So this variant was the one that started out with, I think, in China, and then subsequently it has been replaced by this G, and this G part is supposed to be more infectious but less severe disease. The antibodies generated by natural infection by G or D cross-neutralize, suggesting that the locus is not critical for antibody-mediated immunity. So that should answer that this has so far not been seen to the same antibody, whether do you use against the spike protein cross-neutralizes either the G variant or the D variant. So this is what I came across this thing. And I know there was this talk about A, B, and C, and then there was also K and L. But I have not been able to sort of, I didn't look into it with great detail to figure out what was K, what was L, and those other things.
Yes, Shikha, by the way, it was an excellent talk. And actually, this is my first time to hear something on medical field. The question which I have, when people get tested for this virus, is it based on the testing? It is possible for someone to know whether the person is seriously infected or mildly infected. The degree of infection, is it based on the testing? One can know or not, or just based on the symptom or the degree of illness the person has based from the coronavirus. Only the doctors can know, and not on the testing.
I think the most important thing is the symptoms that you have, whether you have symptoms of mild disease or severe disease. I think that is the most important determinant of whether you have a serious disease or not disease. You can, the PCR, the RT-PCR that we do can give you a semi-quantitative sort of guidance about how much viral load there might be, because it depends upon if you have a very high viral load, then the number of cycles that are required to amplify the virus are lower. So with an RT-PCR, if the number of cycles are low, that means you have a higher viral load, but there is no quantitation as such, available right now. The tests are just reported as positive or negative, and there is no information regarding what your viral load is. And there are no studies that I know of where they have correlated the disease severity with viral load as such.
Yes, Shikha, like my
Okay.
next question, I recently heard that based on the sewage water testing, one can know whether this area is highly infected by coronavirus, because this is the information I got from the CNN news. And do they look for the concentration of the coronavirus in that sewage waste, and can find out how the people are badly infected in that particular area?
I don't think anybody is really doing a quantitative analysis as far as testing is concerned. As I mentioned, the GI tract is also infected by the virus, and can cause diarrhea. And when you test the virus, the virus is present in feces. So I guess that's where the sewage testing will test positive. But I'm not aware of any quantitative analysis.
Hi, Shikha. I want to make two statements. One is it was an excellent talk. Thank you very much on behalf of all of us. This is very important. Listen carefully. If you took chloroquine, if you drank Lysol regularly, and if you took enema of ultraviolet light, you don't need vaccine. You are cured.